Before we can talk about the S phase, or the synthesis phase of the cell life cycle, we need to discuss nucleotide and DNA structure in a little more detail. Now, as a reminder, every nucleotide has the same basic structure. There's a phosphate group, a sugar group, and a nitrogenous base, which just is a ringed structure that has extra nitrogen in it. The phosphate and the sugar is the same for every nucleotide or every DNA nucleotide, but the nitrogenous base is different. And for that reason, they are given different names. So one that has a nitrogenous base that is called adenine, the nucleotide is called adenosine. The thymine nucleotide has the thymine nitrogenous base. The cytosine nucleotide has the cytosine nitrogenous base. And the guanine nucleotide has the guanine nitrogenous base. Now, nucleotides join together through a process known as dehydration synthesis. So, dehydration synthesis produces bonds between neighboring nucleotides. This is done by an enzyme. And so nucleotides, a long chain of nucleotides, also known as a nucleic acid, will be joined together through their phosphate and sugar groups. So it'll be phosphate, sugar, new bond, phosphate, sugar, new bond, phosphate, sugar, new bond. This long straight structure, the part that looks like the back of a comb, or in a ladder, the vertical pieces of the ladder. This part is called the phosphate sugar backbone. And instead of drawing all of the phosphates and sugars and then having to draw the nitrogenous bases, we come up with a simpler way to diagram it, which is just to represent the phosphate sugar backbone as a line, and then to show the nitrogenous bases that are attached to the phosphate sugar backbone. So what I'm drawing down here is the same thing as what I've drawn up here, but much easier to draw. Now, this is a single strand of DNA, but DNA does not typically exist as a single strand. It is a double-stranded nucleic acid. It's a huge molecule. And you've probably seen pictures of it where it looks like a ladder that's twisted. This structure, a twisted ladder, is known as a double helix. Helix means spiral, so this is a double spiral. Now, I've drawn one side of the molecule, or one side of the ladder, but in order to figure out what the other side of the ladder looks like, we need to know something about what's called complementary base pairing. What complementary base pairing means is that the nitrogenous base for adenosine will always form hydrogen bonds with the nitrogenous base for thymine. So if I know that adenosine is on this molecule, I always know that thymine is on the other strand of the DNA molecule. Likewise, if thymine is on this end, then on this strand of the DNA, there will be adenosine, and they will join together through hydrogen bonds. Cytosine and guanine are also paired, so wherever there is a cytosine, there will be a guanine on the opposite strand. And wherever there is a guanine, there will be a cytosine on the opposite strand. So because these they don't match to the same letter, but they always match to a complement, kind of like two puzzle pieces fitting together, we call it complementary base pairing. Now this can be or seem like it's difficult to remember, but I've always remembered it that A and T go together, and these are straight letters, or letters that are made with straight lines, and C and G always go together, or the round letters go together. So if I were to say know the sequence of nucleotides on a molecule of DNA, so I know the sequence of this one. It's TCGACTAACC, right? Then I should be able to predict what's on the opposite strand. So across from thymine goes adenosine. Across from cytosine goes guanine. Across from guanine goes cytosine. Across from adenosine goes thymine, and so on. Right? That's complementary base pairing. And understanding this is essential for understanding the process of DNA replication.